So first of all, thank you. Good morning, everyone, and thank you, Ana Maria and Rafa, for the invitation. This is such a pleasure to be in Madrid and to share with you our work. Ana Maria specified that we need to try to be as conceptual as possible and as broad as possible, because I understand it's a very mixed audience, so I hope I will manage to do that. So indeed, I come from Cologne, from the Max Planck Institute for Biology of Aging, and my lab is specifically interested in mechanistic and evolutionary basis underlying differences in lifespan and aging across species in nature. And we hope that by understanding how different species of animal, plants, and other organisms in nature differ in how long they live and how they age, we can understand something about the way that we age, and maybe we can harness this knowledge to find solutions and interventions. So I will start with a story to keep it conceptual. So this is Abraham Wald. I don't know how many of you know him. This was a mathematician, Hungarian mathematician, that worked mostly in the US at Columbia University at the Statistical Research Group. And Abraham Wald's task was that of helping the Air Force, basically, to minimize damage and optimize investment on repairing aircraft during Second World War. So these aircrafts were coming after bombing with damages, okay? And so the big problem, economic problem, was how can we fix this problem, and how can this plane go back as soon as possible to fly and do harm. And so many people were interested in understanding, oops, sorry, I ran too fast. One of the many most important things to see is where the damage was occurring. So the damage was occurring here, here, here. All these red dots are holes in the planes that were coming back. And so the idea was, okay, we need to patch those holes. We need to shield maybe those parts so that next time that they were going to be hit there, they can come back, but they will not have a hole, so that they can start soon. But this was mostly the intervention. Let's try to shield the planes from the parts that get damaged, at the parts that get damaged. And so the problem is that aerodynamics get changed. Planes are heavier, and so this creates other engineering problems. So Abraham Wald had a brilliant thought, and he said, this is wrong. We should not focus on where we see the holes. We should focus where we don't see the holes. In other parts, we should focus in parts, we should protect parts that are never carrying a hole. And why? Why is that? Because those holes that you don't see are the ones that kill the plane, basically, right? So all the planes that are flying and that are hit in these yellow parts, they will never go back. Okay? And so if you protect the parts that are lethal to the planes, then you will actually save a lot of money, because you will save planes. You will not just repair the ones that are coming back, but you actually keep a higher number of planes. And somehow this kind of thinking of the survivorship bias, so-called, really inspired me. And I think that has a lot to do of our common thinking about how we age and how evolution has shaped aging trajectories in different organisms. In other words, you have mutations, if you want, that are bearable, because these are airplanes, after all, come back. And there are mutations that are non-bearable by selection, and selection actually is very severe and very strict at being careful that no mutation is accumulated in those housekeeping genes, if you want. So how does this kind of scenario, how this way of thinking helps us understand better how we age? And this is how we can think about Abraham Wald's vision on aging and survivorship bias. So let's pretend that this is a genome of an organism, and you have different mutations at every new generation. So what I'm talking about now are germline mutations. These are not somatic mutations that accumulate throughout the lifetime of an individual. But these are germline mutations that occur at every new generation. And they can be green, blue, or orange, depending on when they matter in life. If they matter early on, they will be green. If they matter in midlife, they will be orange. And if they affect late life phenotype, they will be blue. So if you have a deleterious mutation that it's green, that will not allow you to survive to later age, those same variants will not be spread to the next generation with the same success as genetic variants that affect late life. So it means that selection is extremely strong in removing from the gene pool genetic variants that are deleterious, that affect early life. And another way to phrase this same issue is that selection is extremely weak and is relaxed, if you want, for mutations that affect late life. So you see there is this asymmetry in mutations that affect early life and mutations that affect late life. And this translates into a very specific prediction that we're going to be using now for the rest of my presentation. And that we tested in the lab, and I would like to share with you some of the results that we found. Our kind of way of thinking is, if we have a species, ancestral species that has a given lifespan and ages in a given way, we want to understand whether species that evolved from this one, they are longer lived, for example. What are the variants, what are the genetic variants that make these species be longer lived, okay? This is an approach similar to those of us also in the audience that study centenarians, for example, in humans. A little bit different, because we're talking about two different species, though. And then there are other mutations that will make a derived species a little bit shorter lived. So we are very much interested in the evolutionary mechanisms and the forces in general, the evolutionary forces that shaped species to evolve to be longer lived or shorter lived, okay? So this is our fundamental question. And understanding and identifying these gene variants, maybe we can understand better how we age and how different species differ in the way they age. So to tackle this question, we work with different model systems. In particular, over the past few years, I've been focusing on this fish. So this is a turquoise killifish, and it's the shortest lived vertebrate that we know of. It just lives a few months, just 4 months. And why do we study this fish? Well, first of all, as an experimental system, it's great that you have a short-lived organism because you can test many things in a short time. And you don't have to wait too many years, right? So you heard before, mice can live, from Felipe, for example, mice can live years and years, 3 years, and you have these very long-lived mice, and these experiments will last forever. But if you have a short-lived organisms, you can run experiment in a shorter time. So another thing important about these killifish is that they age. They age very similarly to the way we do. They develop cancer with aging, they develop cognitive impairment, they move less, they have senescent cells, they develop fibrosis in the liver, in the gut, and in the heart. So they have many, many features that are shared with us. So that's one of the reasons why we study them. However, they also differ in another part. So they live in Africa, and we conduct part of the work in the lab and part of the work we do in Africa, in Zimbabwe in particular, in the Gonarezu National Park. So once a year, we go in Africa, and we fly with this little Cessna over the park. We identify localities of the park where there is water, and then we drive to these ponds to collect our fish. And this is the typical ponds where we find the fish. Okay? So there is a difference of phenotypes across populations and species that we find in a given pond. And in particular, over time, I've been collecting all these different localities between Zimbabwe and Mozambique together with other colleagues. And what we have discovered is that depending on where you collect this fish, whether it's a very dry area up here or a more wet area where the rainy season is very long down there, you have fish that can be very short-lived, just a few months, or much longer-lived, that live up to 1 year. So this is the same species, and they live short or long. So we can study the genetic basis for long lifespan and short lifespan in nature, okay? And we can find the genetic determinants of lifespan. And please, Anna Maria, can you tell me when I have 2 more minutes to go? Thank you. So we have differences in phenotypes, long-lived and short-lived. And so what we found is that the genetic architecture, so the genetic basis for lifespan differences between long-lived and short-lived populations of killifish, it's very complex. It doesn't mean it's complicated, but it means that there are many genes. There's a polygenic trait, which means that there are many genes, many positions in the genome whose genotype contributes to lifespan and aging differences among populations, okay? So this is kind of, okay, so we have many, many parts of the genome that contribute to differences in lifespan between populations of the same species in nature. So, okay, so that's a starting point. We want to understand what mechanism, what evolutionary mechanisms shape differences in lifespan and that determine the differences in the genetics of these species. And so we use as a resource population genetics. So we know that in the phylogeny of this fish, this beautiful clade of fish, this group of killifish, many times, there was an evolution of short lifespan. It didn't happen just once in the turquoise killifish, but it happened multiple times, at least twice, in this red part of the phylogeny of killifish and down here, okay? So multiple times, independently, killifish species evolved to be short-lived from long-lived ancestral species, okay? And so we asked, what are the genomic bases for this transition? So to address this question, a very talented postdoc in the lab, Rong Feng, sequenced the genome of 45 different species of killifish. And I'm not going to go into the details. And he asked, okay, every time you evolve short lifespan, and this happened here, so in all these species, and here in all these species, but also happen ancestrally here in American species of killifish. So what happened in the genome? Can we see genomic features, genomic properties that are always happening when we have short lifespan, evolution of short lifespan? And the answer is yes, and very obviously so. So what happens is that when you evolve short-lived species, the genome size expands. You have a massive expansion of the genome, almost doubling of the genome. And you will see this in the color code here. So this is small genome size, and this is large genome size. Okay? And you see that there is a segregation of larger genomes that correlates with being short-lived or long-lived very strongly. So even if we take away the phylogenetic component, the relatedness between these species, because maybe all the short-lived species are closely related to one another, if we filter out, in other words, the phylogenetic proximity, we still see a very, very strong connection between being short-lived and having a large genome, okay? So why is that? So first of all, the expansion of the genome happens at repetitive parts of the genome. It's not the coding parts of the genome that expands. That does a little bit, but most of the expansion of the genome happens in the non-coding portions of the genome, in particular in transposable elements. I hope you will hear more about that during the presentation, during the meeting. But in particular, these line elements are responsible for, these are selfish elements that expand in the genome. And these are, again, germline variants. These are not somatic variants. I'm not talking about age-dependent amplification in somatic, an expression of somatic variants. I'm talking about germline variants. So expansion of transposable elements is responsible for the expansion of the genome size, okay? So what we actually were interested in knowing is whether this expansion and also some other genomic signatures that I won't have the time to explain in detail that happen also in the coding portion of the genome were driven by strong selection, if you want positive selection it's called, or purifying selection, or weak selection, neutral evolution, driven by drift. And long story short, what we found, and this is, sorry, I need to mention this. Also within a species, I told you before that we have long-lived and short-lived populations. So we ask whether also within species, this property of being long-lived and having a smaller genome and being short-lived and having a larger genome holds true. And the answer is yes. Even within a species, if we have long-lived and short-lived population, genome size within a species varies based on whether you're long-lived or short-lived. Not within one population, but between populations that are long-lived or short-lived, okay? But the differences among the long-lived and the short-lived populations in a species don't go hand in hand with new genes being strongly selected, for example, in the long-lived or in the short-lived, but quite the opposite. What we observe is that when a species becomes short-lived or when a population becomes short-lived, that happens always when you have population bottlenecks, when you have very strong genetic drift. That means that all the variation of the genes that you have gets constrained by demographic forces. So you have subsampling of the whole genetic diversity, and so you have population bottlenecks. And something like that happens also in our species, by the way, and I hope I will have the time to go back to that. So short-lived populations have undergone very, very striking bottlenecks. Okay, this is unpublished work that we are submitting soon, but this is very, very significant in our hands. So what we see is that when you transition to very harsh environments where the water is available for a very short time of the year, you have genome size expansion. I didn't show you, but also the mitochondrial genome expands. And we think that weak evolutionary forces, like in particular relaxation of selection, leads to the amplification of deleterious variants, in particular slightly deleterious variants. So these are not very strongly selected against. On the other hand, we still see strong selection happening to make such, to select phenotypes that ensure that the species will actually develop very fast and will survive, long drought. However, the genes that are responsible for the positive selected traits are really a handful, while we know that the genetic architecture of lifespan in these species is extremely complex. So we think really that demography and drift drives the shortening of lifespan in the short-lived in these populations. In other words, going back to the analogy I started with, if you have a small fleet or a large fleet, it really determines the accumulation of damage and the damage that you have to deal with when you go out and battle. In other words, small fleets will have still to use the damaged planes. And we think that drift and small populations really affect the distribution of the deleterious variants that affect the aging process. So we don't see much of a trade-off between positive selected and beneficial variants that affect later on lifespan when population size is very small. But this phenomenon of mutation accumulation, of antagonistic pleiotropism, where you have very strong selection for genetic variants that are beneficial for early life and then maybe detrimental later on can only happen when the population size is extremely large. So we really want to underscore the importance of effective population size, whether you are in a mutation accumulation scenario where many deleterious variants accumulate and cannot be fought off against by selection, or where you have a large effective population size that allows to new mutation to be selected. Okay, and interestingly, our species, like I said before, it's extremely striking in this respect because humans underwent a very drastic bottleneck a few years ago, 100,000 years ago, 80,000 years ago. We underwent very, very severe bottlenecks. And our genetic diversity is extremely limited compared to other species, even to laboratory species that we're very familiar with, including mice. So does low drift in human population, has that affected the way that disease-causing variants and aging variants have been distributed in our populations? That's what we asked. So in particular, we asked if genes that are in humans evolve neutrally, so that accumulated these variants due to drift, due to demographic events, they actually are important for aging. So we did, this is a very busy slide, I apologize, but what this shows is that basically you have a distribution of genetic variants in different human populations, Finnish, Chinese, Mexican, these are Bengali and Yoruban African, but we did it in all the sequence human populations. And so there are variants, gene variants that are neutrally evolving and gene variants that are under strong positive selection on the right here. So there are these two tails. And what we see, we ask whether the variants that are evolving neutrally, that are not removed by selection, whether they are enriched for particular Go terms. And the answer is that aging comes out of one of the most relevant, actually the most relevant Go pathway associated with genes evolving neutrally in most of human populations. Another question is whether genes expressed late in life during aging are themselves evolving neutrally. It's an unbiased way to ask the same question. And the answer is yes, at least for when it comes to prefrontal cortex. So we look at expression of genes that are either expressed early on in life during development or expressed later on in life during aging, for example. And we ask, if you are a gene that expressed later on in life, are you under relaxation of selection? Are you evolving neutrally, or are you under very strong selection? And the answer is that this is extremely significant, is that genes actually regulated more during aging are evolving more neutrally than genes that are expressed early on in life. So in other words, demography matters, and it matters to the distribution of the deleterious variants that affect the aging. And why would you care about this concept? Well, our method, basically, of looking at genetic variants doesn't look for variants that are conserved in super long-lived individuals, but we rather look for variants that are accumulated in individuals that have specific type of age-related diseases. And if anything, we're now interested in seeing whether the long-lived individuals are significantly lacking those variants, okay? So it's a different way of looking at filtering genomes. What I showed you is that small population size is compatible with drift, and this, at least in killifish, but we believe also in humans, is very relevant, leads to accumulation of many slightly deleterious variants that are not removed by selection. Genes under drift, are enriched for aging pathways, specifically enriched for aging pathways, and genes more expressed later in life are evolved neutrally. So with that, I would like to thank all our colleagues and collaborators, in particular Ray for his amazing work, and our collaborators down here, and you guys for your attention. Thank you.